In today's video we're gonna be looking at enzymes. What are enzymes? So enzymes are known as biological catalysts. Being catalysts simply means that enzymes are responsible for speeding up biochemical reactions. They create metabolic pathways in which the products of one enzyme are passed on to the next as a substrate. Other than that, enzymes are globular proteins. They are formed by joining together many amino acids in a chain form. Anyway, let's move on to the structure of enzymes. The first very important thing in the structure of an enzyme is its active site. As the name suggests, active site is the part of an enzyme molecules that binds with the substrate during a reaction. Now, let's talk about the overall structure of an enzyme. Enzymes have a three-dimensional crystal lattice structure. This is formed by chains of amino acids. In these chains, amino acids act as the monomers which then fold to form an enzyme. If we talk about the mechanism of an enzyme-controlled reaction, then always remember that there are three important stages. In the first stage, an enzyme with active site that matches the substrate comes closer. 2. The enzyme binds with the substrate to form an enzyme substrate, or commonly known as an ES complex. And in the last stage, enzyme catalyzes the reaction and the substrate is transformed into products. Moving on to the types of enzymes. The enzymes that work inside the cell they were formed in are called intracellular enzymes. An example of this are the enzymes of glycolysis working in the cytoplasm. Type number 2. Extracellular enzymes. These are the enzymes that works outside the cell they were originally produced in. For this one, an example is an enzyme trypsin that works in the stomach cavity. When we talk about the activity of enzymes, there are some factors that we need to consider. Temperature. The rate of an enzyme-controlled reaction increases by increasing the temperature. The increase in kinetic energy increases the vibrations, which makes it more likely of enzyme and substrate molecules bumping into each other. However, when the optimum temperature is crossed, which is basically the temperature at which the rate of reaction is fastest, the enzyme becomes denatured. Denaturing means that the shape of active site is destroyed and so the enzyme can no longer bind with substrate. pH is the second factor influencing enzyme activity. All enzymes work at a narrow range of pH. A slight change in the pH can have a huge impact on the rate of an enzyme-controlled reaction. For example, the pepsin enzyme works in acidic mediums where you can find low pH values, whereas trypsin enzyme works best in alkaline mediums where there is a higher pH. Substrate concentration. When the enzyme molecules are available in a reaction, an increase in the concentration of substrates will increase the rate of reaction. However, if the number of enzymes is kept the same and the concentration of substrates is kept on increasing, a point will be reached where further added substrates won't increase the rate of the reaction. This is because there won't be enough active sites available to bind to the substrates.
We've lost track of time, don't know how to feel, but I'm not sorry.